Hello. Uh, Today we will explore linear mixed effect models and their usage in psychophysiology. The basic idea behind this model is to analyze data that comes from repeated measures or hierarchically structured data. Uh, in psychophysiology, it often means measurement taken from multiple participants across sessions and conditions. Yeah. In the context of this paper published on 2008, the authors discuss common experimental design in psychophysiology. So the data include multiple observation from the same participant and their EMG responses to various videos. Yeah, so it creates a closed structure where both participants and the videos could be treated as random effect. However, given that the appendix of this paper only shows the measurement of EMG for each condition, so we cannot use item as one of the random effects. So in this practice, we will just consider subjects as the source of the random variables. Okay. Yeah. So also particularly this data set, this paper describe an advantage of linear mixed effect model. That is, if you read through the this example, yeah, there is another part of the analysis when 12 subject has missing values in either one or two conditions, usually in repeated measurement experiments, when subjects cannot provide every measurement across experimental conditions. For example, in this study, there are 4 treatments. So maybe subjects EMG sensor might lose or just do not respond during one of the conditions, and then in under that case, a given participant might just have 3 measurements but not 4. So usually in convention analysis, we will just remove the old data from subject with missing values. However, in this paper, it's mentioned that since mixed effect model can use all available data without the need to exclude participants. So yeah, in the case with missing data, linear mixed effect model would make the analysis more efficient. Okay, so now in in this R code, we will walk through some exercise, yeah, to analyze the EMG data from the paper. So in the first step is load the required library. So we load 3 R packages, LME4, to perform linear mixed effect analysis. LMER tests. Well, actually, I will use LMER method in this library. So I just keep my convention behavior. So I always load two library, LME4 and the LMER test. Yeah, but in the following analysis, we load LMER test after loading LME4 package. So we will use LMER method from this package. Yeah. Okay. And also we use this LATTICE package to for visualizations. Okay. So after loading required libraries, we set the working directory to locate to the location where we keep the files. And then we load the data using read.csv method. So and store the data in the data variable. Okay. And after load the data, here I specify that the emotion column is factors. So it will tells R to treat information in the emotion column as as categorical variables. So it is important because we are interested in comparing the effects of different emotional conditions. Okay, so now let's go through this processing step. Okay, uh, now we, uh, we can visualize the data. Okay, so BW plot creates a box and whisker plot of EMG amplitude across different conditions. Yeah. So it helps us to understand the distribution of EMG signals for each conditions. Okay. And for regarding the XY plot, it generates a scatter plot of EMG amplitude for each subject across emotion conditions. So yeah, this 1 over subject, oh here, sorry, here, yeah, so here this emotion over subject means that we create separate plots for each subject, so we can see the individual differences. Okay, you can notice that for some subject there is no much change regarding the type of videos. However, some subjects shows different EMG activities in response to different videos. Okay. So now after visualizing the data, now we use LMER method from the LMER test package to fit linear mixed effect models. Okay, so yeah, so the formula indicate that EMG, the information in the EMG column is the dependent variable, and emotion is the fixed effect representing the different emotional conditions. And here the parentheses indicate a subject is the source of random effect. Yeah, and so it will, and we only consider the random intercept model. So it will account for individual differences in the intercept. Okay. And here the argument REML equals false specifies that we use maximal likelihood estimation instead of restricted maximal likelihood. Yeah. So and it will allow us for comparing models with different formula in the future. Yeah. So and now we can take a look about the results. So the summary function provides detail of the output. So we can see that there are three information regarding the effect of fixed effect. So you may wonder why there are three rows here, because there are four categorical types in emotion conditions. So in this case, we need three contrasts to describe the structure of to describe the differences between four conditions. Okay. Yeah. So and now let's go to the next part. So you might wonder how to interpret this emotion POSI, emotion relax, and emotion sad. So this bicoding contrast method, yeah, uh, we can see the default coding schema for the emotion vectors. So yeah, so it will indicate that for one of the particular condition, for example, this AGITA, this name, what was is considered as the baseline conditions. Yeah, so then you can so in and the first row in the output of the fixed effect here emotion POSI shows that this it's this factor the first row is a summary of the difference between the first two conditions. So we use the condition labeled as 1 to subtract the performance of the condition labeled as AGITA. Yeah. And so then the next row of the fixed effect emotion RELAX indicate the contrast under this column. So it indicate that we're looking at the difference by using relaxed condition to subtract the data from this AGITA conditions. And yeah, so so this is the default coding schema in while using linear mixed effect models. Okay, and we can also evaluate the output of the random factor after fitting the models. Yeah. So we use dot plot to visualize the random effect or the subject level variability. So in this case, we only consider the intercept as the major source of random variable. So it will help us to see how different subjects vary in their EMG responses. Okay, yeah. So also, yeah, this variability is independent of the emotion of the fixed effect. Okay, and now I want to do another demonstration that is in addition to use this exciting similar condition as the baseline, we can use relax as the baseline conditions. Yeah, so you can, well, the reason is that, well, relax means subject is in the neutral conditions. The other three conditions, in the other conditions, subjects will see a video with some kind of emotional content in the videos. So we can use the neutral condition as the baseline and uh, and use the rest of us the three conditions to compare with the baseline conditions. So in order to do that, we have to create our own contrast. So here we define a custom contrast matrix for the emotion factor. So the matrix M sets up the specific comparisons between the levels. Okay, so let's evaluate this method, this script. So now you can see that uh, the the output of contrast is different from the original message. Okay, so here I try to make the name of each contrast more like easy to be comprehended. So the first contrast is the exciting condition minus relaxed conditions. So by uh, looking into the detail of the matrix, you can notice that the third row relax has the value in the third row relax are zeros. So it means now this level is treated as the baseline. So in the first contrast, the AGITA is coded as 1. So it means that in this first contrast, we try to compare the EMG response to excitement video versus the relaxation videos. Okay. And in the second column, it means we create a contrast by using EMG value recorded in the positive condition versus the EMG responses recorded in the relaxed conditions. And the third contrast indicate that we built a contrast by using the EMG responses to set video versus the EMG responses recorded during the relax videos. Okay. And now we built the model again, and here you can notice that the first contrast is statistically significant, and the second contrast is not significant, meaning that, well, there is no significant difference between EMG response to the positive video and the relaxed videos. And the third row indicates a significant difference between the set condition and the relaxed conditions. And in the first column of the first row, in the first column of the fixed effect, you can notice that the value, the coefficient value are all positive. So yeah, it means for the under the first and the third conditions, the EMG signal is significantly higher than the EMG signal to the relaxed conditions.